Hello, Jesse.
Morning. How are you?
All right. How are you?
Okay. I may not be able to talk that much later, but I'm just stopping by.
Good. Yeah, I mean we have a pretty full slate today. We'll see who
Yeah.
shows up. Let's wait for a few more people.
Has there been any? I know I wasn't really here the last 2 weeks. Has there been any movement on the Docker-related things? I'm looking at the to-do list for the passport.
Yeah, not real. We haven't talked about it. We kind of talked about it we reviewed the board a little bit, but that's about all we mentioned on it. So yeah, I mean that's like one of those things where you kind of create some content around it. So there are two parts to it. So putting it in the Docker container itself, but then also creating the thing that goes in it. And so, well, actually, Ujjwal has a simulation. He's doing his 3D Blender now. He's working in Blender. He's doing a simulation of the worm. So I don't know if he'll update us today on it, but I think it was in last week's meeting.
Okay.
Hello, Ujjwal.
Hello, sir.
How are you today?
Fine, sir. How are you?
All right. So I guess I don't know who else is gonna show up to the meeting. I know Dick is taking alternate weeks off this summer. He's going out into the woods and fishing opportunities where he lives, and so they don't have very long of a summer, so they have to take advantage of it. So he's going to take every other week off. So I guess we'll hear from Mayukh and Ujjwal on their GSoC updates, just the weekly updates. Give a quick update if you have demos, show that. If you have questions, raise them. And then I think Krishna was supposed to show up and give a talk on his idea. I don't know if he's going to do that yet. He's still going to do that. And then I was going to give a talk on this Euler cycles, multi-cell systems stuff that I've been working on. Oh, there he is. And then, yeah, that should be the meeting. So, hi, Krishna.
Hello. Hi, how are you?
All right, how are you?
I'm good.
Are you ready? Are you going to present today, or?
I can present,
Okay.
but okay, I can present.
Well, yeah, not right now. We'll do it in a little bit. I want to do the updates first for Mayukh and Ujjwal, and then we'll have you present. Okay. So Mayukh or Ujjwal, who wants to go first?
I can go first.
Okay.
So like this week, I have like started the coding period. So in the beginning of the coding period, I have started to work on the things like reducing number of steps to reach to the model. So I have done that basic architecture changes. Also, I have started to create new things from from keeping the design perspective in view, and I have tried to make things more and more clean and easy to understand for the user. I have pushed some code like for the layout and for login purposes. So I have created that, and by right now, by the end of the day, I am going to push almost all the front end part. Then this week, I'm going to integrate the front end part with the Django platform and start working on the React.js code dashboard.
Okay, that sounds good. What are your your long-term plan? You're going to stick to your schedule, and you're working on it. And what is your you ever think you anticipate any problems with your schedule or keeping it or?
Right now, I'm strictly following the schedule which I have given in the proposal, and right now I haven't faced any problem yet.
Okay.
But I think the time can be consumed in the segmentation part, like Basileia segmentation. Oh, sorry, e.elegant segmentation part. So I'm trying to come up to it as soon as I can.
Okay. Well, I mean, I just want to get a sense of where everyone is each week in terms of their schedule. So, but no problems. That's good. All right, Ujjwal, or thank you, Ujjwal. That was good. How about Mayukh? Can you go for a
Yeah,
short?
am I audible? Can you hear me?
Yes.
Can you hear me? Yeah, so let me just share my screen real quick.
All right.
Let me video. Yeah, can you see me?
Yes.
Yeah, I was already actually talking to Bradley about this thing on Friday. We were talking, right?
Yes.
About this epic data set. There's this huge data set full of videos of worms and full of metadata, which is like time tagged. So it's really great for our purpose. So then I started, first of all, it took me some time to understand what's going on in the data set, but then this diagram kind of helped. Like what happens is that the initial cell, like the initial embryo, it gets split into n embryos in such a process that it can be tracked back to its ancestor. And somehow each of these little small cells at the bottom can be tracked back to the ancestors from their properties at a given time. Okay, so suppose then I moved on to like visualize the data. What happens is that these cells, these ID cells actually move through the embryo as time goes. So there are x and y coordinates, as you can see, like this is the raw data. I just gave four column rows. You can see x, y, z, and all of that, like positional data. So I just plotted the x's and y's for a few of the cells, and then I found like these kinds of movement, unique movement that is distinct to each type. So then, like moving on, I kind of like after this, I just took a smaller, like not really small, like the whole thing has hundreds of CSV files, but I took like a few of them, like 3 or 4 of them, actually 4 of them. Then I tried to train a deep learning model on it. So, but before that, the dataset was, as you can see here, it was highly imbalanced. The classes were very imbalanced. So I just oversampled them for now because I don't know how to really augment by adding noise and such. I'll experiment with that soon. So I just trained a model which can like it takes 4 parameters. Parameters like cell, wait, hold on. It takes parameters like its position, x and y, its size and its time, and then it gives back a label which corresponds to its like compact cell name. Now what is compact cell name? It's like you can see that this diagram. Wait, let me show you. This diagram, this can like if we extrapolate this one, we have like 700 or something, like a huge figure, a huge number of cells, like a huge number of labels, and they are highly imbalanced. So what I decided is that I just took the ancestors as the label. So I took the compact symbol. You can see it in the diagram. Like if the original name is a longer name, then we just take its ancestor's name as a compact name to like reduce the number of labels, which would be useful in such a dataset where it's highly imbalanced. So then I trained a model and I got around 90. Like for now it's fine. I'll just have to tune it and I'll get more, but that's a different story. And then what I was doing today was that I then moved on to like do some principal component analysis. What it does is that it kind of like reduces the dimensionality of the data and it tries to visualize it in 3 or 2 dimensions so that like we can make a little bit more sense of it. So I just took the same data set and I did some PCA, and like as you can see there are a lot of labels. These are 32 labels. And you can see it's like a very messy scattered plot, but still you can kind of like, it's hard to make sense of it. But then I made I spread it out in all the other, like I just plotted them individually, and you can see kind of like, it's just interesting to see how like the similar ancestors have like similar patterns in these parameters, like PC1 and PC2, like they have the principal component 1 and 2. Yeah, so this is what I was doing.
That looks good. So this is PC1 versus PC2.
That looks good. So this is PC1 versus PC2. Yeah, PC1 and PC2 are generated by the code itself. They're not like, they are just dimensional, like there are 4 parameters. I'm just reducing them to 2 parameters so that like how should I put it, like it's like a 2-dimensional projection kind of a thing of the 4-dimensional data. It's like a projection which kind of gives us an idea of what's going on. It loses data. It obviously cannot like project the data in 2 dimensions in 100% accuracy. It obviously loses some data. Like in this case, it can only show 70. It can only retain 70% of the data when it's reduced to 2 dimensions. The rest 30% is lost. So like even with 70%, kind of gives us a good feeling about what's going on inside. And you can see the ones which are starting with C are like positioned and they have a similar shape and a similar size. And the ones which are A have a similar shape and a similar size. And all of them have a common center anyways.
Right.
Yeah, so that is what I was doing.
So what were the 4 parameters you used?
Parameters you mean? Okay, the 4 parameters, yeah, wait, hold on. The 4 parameters were cell, like, wait, 1 second. The 4 parameters. I'll just show you. Yeah, the x coordinate, the y coordinate, the time and the size. These are the 4 values I used. Yeah, so that is what I was doing. For now, like, any suggestions on what are the other parameters that I can use which would make it better? Like you can see the raw data here, right? What are the other parameters can I use? Like there is this thing called global, local, blot. I don't know what they mean.
Blot is probably like a technical term for their fluorescent stain. Global and local, I don't know what those are, but it looks like local is the same as
this thing called GMAD.
You can try out like great search theory for that. Like if you want to use the best parameters, so what you do is like it will compute all the possible parameters for it, and you can get the best result out of it.
Yeah, I would just not worry about
I'll do that.
or cross or blot or any of that.
Like scaling it is going to be pretty easy. For now, it's just I'm prototyping right now.
Right.
Hello.
It's just like being constructed. We can change them.
Yeah.
You can experiment with the p values. Like when you are doing the PCA, you can change the threshold value of the p. Am I audible?
Yes.
Yeah. So you can shuffle the threshold values of p, and you can just like you are sorting out the attributes, right? So you can change the value of p, and you can see how your model is getting affected. But it follows the Huygens principle, like the curse of dimensionality. More the dimension, it will get harder to get a good estimate model. So you can you can find the optimum number of attributes. They should not be very small, like 3 or 4, or not like very big. So you can settle for an optimal number of parameters. You can
Experiment with that.
yeah,
I
you can
think
experiment with the values and get it done by that.
I think like Mayukh is trying to identify like first like which parameters are going to give him the best values out of the data set. Am I right? You want to experiment with the
Not
parameters?
necessarily the best value. I just want to know which are the parameters which would be used, which are practical.
Yeah, that is
Which are practical because
into the play. Hello.
Usually people will plot P1 versus P2 to get a sense of the variation. Usually, if you plot both of those axes, they should explain the majority of
P2 are like abstract. They are two abstract parameters. We just don't know what they are. They're just like projections. You can consider this plot as a projection of what's going on the higher dimension. Yeah. So that's pretty much it. This is just some analysis. This is not like to trade something or anything. I won't use principal components for trading because it leaks a lot of data. It leaks 30% in my case. Like it loses 30% of the data when I reduce it to 2, which is like pretty a lot. And I want to use more while training. Like as you can see, I was using only 4 of them. But I want to use, wait, let me show you. I want to use almost all of them. But first I want to understand what's going on because my next step would be normalization. As I was talking to Bradley that day about normalization using Z transforms or things like I'll see to that. But I don't know about these blot and cross values, so I'll have to study about what these are. I'll have to read more probably, so I'll know whether or not to normalize them or not. Because it will be better if they're scaled. It will train much better if they're scaled to a certain like -1 to +1. I don't know if like scaling them would make sense. Yeah.
Well, yeah, we can talk about that. Yeah, I mean, people use eigen values for that as well, like for. Yeah, we can talk about that later. In fact, I might give a talk on that soon, like the different multidimensional methods that people are using for biological data. Comment on the data here. So the data table in out 0 up here. Yeah, right there. So I think the parameters you want to stick with are time, cell, and then the XYZ variables, and then the size. So the rest of them, I think, are like...
No, it's actually... Yeah. It is identifying cell.
Yeah. The rest of them are kind of, I think, technical parameters, so it's not something that we would find useful. But, yeah, I think that's good. And I think that's a good way. I would add in Z as well, although I know that the variation on Z is not as great. So, that's not... gonna predict.
Yeah, for now I'm ignoring, but it's just adding one more column. I can add Z at any time.
Yeah.
It won't take much effort. And like for now, the model is like the input for the model is like these values, and the output is the cell's ancestor name.
Yeah.
Like whether or not it's from an ABA or some P or whatever it is.
Yeah.
Like from this flowchart, like it can look at one of the cells and its position and the time value, and it can tell whether or not it belongs to which compact cell name it belongs to. That is, to which ancestor does it belong to, or to which lineage? I guess that's the word. I guess, right?
Yeah, lineage. Yeah, lineage actually, but But of course, there's one problem you're going to run into, or there's a problem with that is that there's like you're averaging over the entire lineage. So sometimes we think about it in terms of lineages and sublineages, but you're combining a lot of data from different time contexts, and eventually they become functionally differentiated, and so it's a little bit hard to pull out a lot of the variation from that. So we might try different partitions of the data as well later on, but just get that working. That looks good.
Yeah, different time slices partition?
Well, yeah, I think at different time slices you could...
No, I mean different time slices. Do you mean that when you say partition?
Well, I mean so you see these cell names?
Like you can start like I was reading a paper today, and there it said that they stopped collecting the data after the 350 cell stage.
Right.
After that, because it diverges too much after that.
Yeah.
So should I stop at the end cell stage to make it better, like reliable?
No, probably go through the entire.
It won't have a large input domain, but it will be reliable.
Yeah. Well, no, go through the entire data set. You have, I mean.
I'll just send the link tonight. And you.
Yeah. What I'm saying is that you're, you know, when you get from like, say the 256 cell stage to something larger, or even like from the, 64 cell stage to something larger, you get things move. You get things that are starting to differentiate. So, it might be worth. Well, we can talk about that later, but suffice to say, when you look at a sublineage, you're getting a lot of averaging effects over time. But this is good. So if you were going to push some more stuff, keep pushing stuff on this to GitHub. And then how are you doing in your schedule that you proposed? Are you running into any problems with that? Or?
No, for now there are no problems, but there is one minor problem, that is that work will slow down for a bit because I have my exams coming from next week.
Yeah.
So next week would be a bit slow. And I'll study for the coming week. I won't work much. I'll be working, but it won't be like the last few weeks. So that's the thing. Yeah.
That's good. I mean, I just wanted to check in on the schedule to make sure that, like, you didn't think that something was insurmountable. Again, like, we'll do this weekly, and if there's nothing to report, there's nothing to report. But please, if there's some problem, and this goes for Ujjwal as well, please let me know as soon as possible if you have problems with computing resources or something running. We can either I can help you, or we can just put it aside for a while and then move on to something else, just so we keep the summer moving and we don't have a bottleneck where you can't overcome it for a couple weeks. That's what we want to avoid. Ujjwal, did you have anything to demo or?
As of now, like I have already demo you what we are working on as a DevoWorm AI portal, so it looks pretty similar right now. Also on the blocks part, right now my portfolio doesn't support block system, but I have started making it, and I will be updating all my 3 or 4 weeks block by deadline.
Okay, that's good. Yeah, drop me the links in Slack when you can. Thank you. Thank you for Mayukh and Ujjwal for the updates. Looks like you're doing pretty well, both of you. Krishna, I'm gonna move down to you, and you wanted to present on your idea that you have for a project.
So okay. Is my screen visible?
Yes.
I can see.
So, okay. Hello, everyone.
Hi.
So what I am proposing is to make custom-made convolutional neural networks for biological data. So as we all know that CNNs are used for deep learning in case of when the input is an image. And so why we use pre-trained models? Because pre-trained models are, they don't start with exact zero. They have some base, they have some weights already being learned, and when we retrain them, the task of training becomes much more easier. So the benefits of pre-trained models that they have leveraging, they are super simple to incorporate. They don't require much labeled data. They are versatile to use. So the main thing, like most of the CNNs are trained on ImageNet. As you can see here, the classes of ImageNet are like car, cup, plate, and train example. But the biological data, it is very much different from the things that we have in the ImageNet. Am I audible?
Yes.
Yeah. So
Yeah.
like yeah, if we have like slide of in cell, it is I think 360 degrees different than a cup or a plate. So what I am going to propose to do, I'm going to yeah. I'm going to custom make an CNN which is not trained on ImageNet data, like all our pre-trained model, ResNet, EfficientNet, VGG, all of them are usually first trained on the ImageNet data, then they are used for other things. But what I'm going to do, I'm going to customly make an CNN, and then what we are going to do, we are going to train it on specified biological data, whatever we find, like we find cells, we find we find cells of plant, we get some plant data, we get some biomedical image analysis data, whatever we can find, like X-ray images, MRI images, cell digital pathological data, whatever biological related data that we have, and then we are going to train it, and then we have to make two feature extractors. So the reason I'm proposing for two feature extractor, because for example, let us consider ResNets. ResNets have more than four variables, like they have ResNet 50, ResNet 101, ResNet 18. So what we can do, we can make two models, and one that is for state of the art results, that is having more convolution layers and which takes more data to train, and it's heavier model, and one we can make a light model for a mobile model that has fewer layers, but which acts a little bit fast than our main state of the art result giving model. So any questions?
Oh, go ahead. Okay, I'm gonna.
I have a question.
Yeah.
I have a few questions. Yeah. First thing is that what will the model exactly do?
It will extract features like ResNet. It would be a general
Extract features.
Yes.
You mean will it label data? What will it identify or what will it determine?
So
it will take images of X-rays, all sorts of things, right? But what will it identify?
So it won't identify anything. Like for example, let us have the example of ResNet. ResNet itself doesn't do anything apart from extracting features. But when we use it in UNets, it's used for segmentation. Like our model will get an image and it would output an image and learn the features. But it would be having its its application in segmentations and in for other like semantic segmentation and for other things. It would be just like a version like ResNet. Like for example, let us take the consideration of UNets. Like UNets itself use ResNet in it for feature extracting. Same would be done for our model.
Yeah. So have you thought about the data?
Yeah, I will be. I've searched on Kaggle and on Google Open data sets. Biological data, is very much restricted because of privacy concerns. But we do have sufficient amount of data, and using for example, bio data, which is I think considerably less than the ImageNet, but since it is but since it's more like connected to our further which will be again given, so it would be better to use cup and plate for training for predicting cancer than to be taking images of plant data or any other MRI for predicting cancer. Like so, analogy here is that we are hello.
There's already a Kaggle data set or the cancer data set?
Yeah, I'll be taking cattle dataset. No, not only a cancer dataset, whatever I can find, whatever has the license that I can be using it. So I can take from Kaggle, I can take it from RSNA, like Radiologic Association of Northern America. I can take it from some government agencies, whichever of them will be, providing us the data. So there is ample amount of data freely available on Kaggle and on open Google datasets.
Yeah, so how do you plan to train it? Like, is it on Kaggle kernel or Google Color? Like, it will take a lot of computational power.
Yeah, they won't be able. Yes, they won't be able to, provide me the computation. It would be, as per my estimation, it would be taking at least 1 to 2 weeks of training on a good GPU, for example, like in Volta V100 or RTX 2080 Ti. So we can, ask for cloud credits since we are doing it for an open source. Might be Google or other, like we have some GPU providing service papers, Cloud Hub. They might
Like,
be.
as far as I know, AWS gave like cloud hours they gave, I think. If you have a student account, they give, yeah, we use
But
AWS.
they will also giving us. Plus, we can ask from Google Cloud Services that whether they are willing to, kind of fund our this project. Then we have Paperspace, Floyd Hub. I think someone would be, really happy to provide us with the GPU. Or what we can do, we can contact any research institute like some university or that, and then I can take the access of GPU from SSH and all.
Yeah, other than that, you can just, if we don't get anything, what we
Yes.
can do is just checkpoint the model.
Yeah,
You can just train it for a little bit and then train it for a little bit again.
yeah, but
So
that would be a tedious process. Like it would take months, literally 1 or 2 months to do this. But I'm pretty much sure that we will get GPU credits for free from some of the service providers.
I think the biggest hurdle would be data.
No, I don't.
The biggest hurdle.
Yeah. No, I don't
It
think
will
that
take time to
data.
get
No,
good
I don't
data.
think that it would be a really big hurdle. Because, for example, like I'm in touch with a few agencies. Like first of all, let us take the example of RSNA. They have all the radiologic data freely available. They have more than, I think, half a million of images. Then I can get further data from various agencies that I'm also in contact with the agriculture, university, a few agriculture universities of India. They are also having some plant data.
So it's like your model wants to do a lot of things, but is it not more rational to make two? Like suppose your model, you make a huge model. It can analyze radiological data and also it can analyze plant cell. Okay.
Yeah.
But is it not more rational to make, because you were complaining about the size, so is it not more rational to make a smaller model like a Resnet 18 instead of a Resnet some bigger value, like make a Resnet 18 instead for plants dedicated and one for radiological images, like specific
No,
models which are with the
that can be further than I am making two models, one smaller one and one bigger state of the art result giving model. So
no,
what
I
I'm
mean
doing, I'm making a general model for biology. What we are doing here, we are fragmenting it into like for zoological data or botanical data. So what I'm making a general model for biology. General models of biology don't exist, right? So we can further devastate it, but as per now, my vision is to make a generalized model for biology, which covers all the aspects, right?
Yeah, I have a couple
yeah,
questions.
great. It's actually great.
Yeah.
Just keep me updated.
So yeah, I have a couple questions, and they kind of lead off what Mayukh was talking about.
Yeah.
So, and the first one is you have like all these different systems. You have like like plants, animals, radiology images, which are animals usually, but they're different in their scope. You're going to find a lot of similarities, but you're also going to find a lot of differences. So, I mean, one of the things we have is, people think about this when they think about like the tree of life, where like when people try to develop very broad theories in biology, is that they have a lot of variation, and it's not all the same in the sense that we can really understand it using a very simple model. So I don't necessarily worry about that. I think it's something we'd have to account for in terms of like feature selection. So you're going to have like a bunch of features maybe that are similar for the different categories, and then some that are going to be like different for different categories. So you might have a cluster of features that are like something that's shared between radiology and plant data, and they might be like technical artifacts, or they might be like actual things that are just kind of that you're able to see in common. I mean, I'm talking very generally now, but I think you probably see it from the data. It would emerge. But then you'd also have things that are shared or that are not shared, that are unique to, say, like animal data for cells. If you're doing cell image data with a microscope, you'll find stuff that's very unique to that. And so, I mean, that's something you want to take into account. I do kind of worry too about what Mayukh was saying about like the size of it, at least in terms of like, well, we talked about this before about balancing your classes, about getting like the right, getting things that aren't just kind of spurious things that you identify, like I talked about technical artifacts, but also just kind of like things that we already knew existed, like this is a cell, and then we already knew that was a thing. I think though, so that's an interesting approach. You're doing feature detection, so you're just basically, when you segment an image, you're delimiting a thing. You're just drawing a circle around it or something. You're pulling it out and you're saying this is a thing, as opposed to just picking at random or just like kind of trying to draw by hand, because that's what people do in a lot of the microscopy, like some of the microscopy analyses that they've used in ImageJ, for example, or they'll use algorithms, but people will use like hand, drawing by hand, or they'll use like templates, and so it's not. There's a reason why people turn to segmentation. What you're saying is you can pick features like the way we do segmentation, but it's like you're picking the features out. Kind of right.
Like plant and animal data are different. They are unique. They are different in many aspects, but they are same in many aspects. Like hello. So if if you are doing a multivariate analysis of the data, they all are unique. They are different in many aspects, and they are same in any aspects. So what we are going to do, we are making a general model, like So the reason I am taking both botany related data and animal related data is that I want it to be a generalized model. Like for example, if I'm going to train it, pre-train it on something like cups, plates, and bicycle and car, it's more feasible to train it on so the things which are more related to the data. Like, so if we are going to make a general feature extraction, it should be having all kinds of data. Like if I'm giving plant data also, then it would be much better in extracting plant data. If I'm going only for animal data, then it would be have some kind of bias. So what I really want to convey, like I want to make mix 50-50%. The exact 50 is not really possible, but I want to make the classes as much balanced as possible. Like I'm going to take 10% of data radiologically. I'm going to take 10% of data from plant cells, and plants also have like different regions. For example, that if I'm going to take a leaf cell, there would be a balanced, systematic incorporation of data here. So I'm going to when I'm going to merge data, I'll be making sure that every kind of data is in proportion.
Yeah. Yeah, I think that's good. And then you might also do this like initially balance them by their type, but then once you know like some of the features, you might try
Yeah,
like rebalancing them. Like now
yeah,
that you know like some types of radiology data are maybe closer to
yeah,
plant data, um, because they do this with like phylogenies all the time, and I think I've talked to people about what those are. Those are trees where they try to figure out how things are related in life. So like
yeah.
plants, animals, bacteria, and a lot of times those are very contentious because they'll find features and they'll use them to like build a tree, say build a hypothesis. This thing is a common ancestor of two other things because it has this feature that it looks similar, but it's changed over time. So but the thing is that they constantly update these trees based on different criterion that people use. So people have incorporated gene sequences. They've had different looking trees. They incorporate different morphological traits. The trees change again, and and they build consensus trees, and they're different even still. So I would expect a similar problem here, where you'd have just depending on your input data, you'd have constant like your proportions of your features would change. You'd be pulling out features, so you at least know they would exist.
Yeah. And
So yeah,
no matter how different plants and animals are, they are relatively more relatable to each other than a couplet and a plant or a bicycle and a cell, right?
right,
So yeah.
yeah,
So the things that you are saying, they are 100% true. But we can at least come up with something that is even 1% better than the existing one, then it would be a great achievement, right? We can at least try to make things and see where it leads. Then we can subdivide it into specified model just for botany related data, right? For plants and all. Then we can further divide it into animal and in animal like radiological data. So we can further bifurcate and make different models, but we can start off with a generalized model.
yeah.
So we can then see where things lead.
That's good. Sounds good.
Yeah.
Jesse or Ujjwal, did you want to ask any questions about this?
I think like the idea sounds nice, but the
Thank you.
problem is going to be like in biology, like what you're talking about, like bicycles and all these things. So these are something that are very I'm missing if I'm not missing the word, like very robust in nature. Like a cycle looks like all the time what a cycle is. But in our case, like if you are treating cells itself change shapes a lot of times.
Yeah.
This is something I think we have to tackle about.
Yeah, yeah. That's what. That's why I'm saying that I will be taking every kind of cell available to me as in data, right? If I having hello.
Yeah,
Yeah. So if they are like if I'm taking every possible kind of data that can I have in an in a good proportion, then I don't have the problem of it being overfitting, right? It would be biased. Like if I'm going only showing it a photo of a cell type, like for example a round cell, that or the structure is kind of round in nature, then it would produce biased result. But if I'm showing it everything, like if cell changes its shape, doesn't matter because I'm showing it all the possible shapes that are available. So wouldn't it be? It would. I think there is a message from Jesse.
yeah. I'll get it in a minute here. Finish your
Okay.
thought.
So if I'm showing it every possible kind of data, then the chances of bias are reduced and are significantly reduced. If cells change itself, doesn't matter. It would be a concern if I'm only showing it round cells and then cell change its shape and become kind of square. But I am showing it square, round, oval, every possible thing that I have. Then the possibilities of what you are saying are, I think, significantly reduced.
Okay. Yeah. Yeah, so you would train it on like basic shapes.
Yeah, I am training it every possible thing that I have, like every possible thing that I can give it for learning,
Yeah.
so that it is sufficient in extracting features of possibly every possible thing that we have.
Yeah.
So more the data, better are the results. So more the diversity of data, better kind of result. So if I'm teaching that network every possible kind of data, then it's not cramming things of a particular type. Like it is having more flexibility to from radiology to pathology to then plant related data. Then it can perform equally when in all kind of situations.
Yeah.
And when you're teaching someone about bicycle and then you are showing him a photo of a cell, it's I think worse than teaching someone about a plant cell than showing him an animal cell. There is somewhat analogy between living things, uh,
Yeah.
which yeah.
Yeah, I think that's a good observation. Yeah, I think definitely. Yeah, we'll talk about this in coming meetings. We could maybe if you can write something up like a short outline and maybe make some sketches of what you're thinking in terms of like the input, the training data, the training data for shapes and that.
Then I have to custom make the CNs. I have to decide how many layers to put, what should be inside those layers. Like so, with the coming meeting, we can make a draft of things to be done, and the progress is like
Yeah. Yeah. Okay. Yeah. So yeah, try to work on like a next step would be to work on an outline of what you would do and the steps and all the things you need to do because you need to have all you want to have like a list that you can check off and figure out where
yes, sir.
things are. So thank you very much, Krishna, for that. Let's see. Jesse had a question. It wasn't about this, but it was about. I have some questions about the basic non-neuronal cognition papers, but I'll probably ask you for that in Slack. Yeah, please ask me about that in Slack. That's something I think we talk. We've been just kind of talking about that they exist in the last couple of weeks, but we haven't made great strides in that area. So in the recent past. So we'll probably turn to that in a while, in a couple weeks. But please ask me questions about it, Jesse. I think that would be an interesting conversation. So I'm going to present on this stuff for the last 15 minutes. This multi-cell systems thing. Let me share the screen so you can see my presentation screen here. You can see my tab here with it's not up yet. Here we go. You can see that. Okay. Let me put it in present mode so we can. So this is some work that I've been doing over the last couple years not in very close collaboration with anyone, just kind of like putting together some ideas on this if it ever loads. There we go. It's called Euler Cycles for Life. That seems like a little bit of a weird title, but I think you guys are familiar with the traveling salesman problem. And that is this image up at the upper right, the Königsberg Bridge. So the idea was there was this town in medieval Germany where they had to figure out the minimal number of bridges they needed to cross these rivers and link the town together. And the Königsberg Bridge problem sort of optimizes for that situation. And that's a bit applicable to a wide range of problems, not just this toy problem, but a lot of graph theory uses the TSP, and as we'll see, there are other domains as well. And I show you these pictures on the lower right, these seashells and diatoms, because these are the types of organisms where you have the types of morphology I'm going to be talking about. but it's a kind of a sort of a very general analogy. So here are your Euler paths. So Euler paths also can be used to define networks, but also geometric networks or geometric like shapes or complex geometric shapes. So here on the left we have an octahedron and a dodecahedron. So these are with 8 and I think 20 sides apiece. And so these where you have these cycles, inside of each of those cycles is a side, right? And so you count up, this is 8, and I don't know how many are in here. 6, 7, 8, 9, 10, 11. It's like 12 or, yeah, it's, anyways, the point is that, each of these cycles represents a component, right? And so you have this network, and you see this kind of even in Volvox colonies where you have a bunch of cells in this colony, and you have certain properties of shape that require, well, they don't necessarily require optimization, but you end up with this optimization of shape. And you end up with, but importantly, you end up with these sort of almost like networks of things. And when you have this multicellular structure, you also have networks in terms of like functional networks. So when you have a single cell, it can interface with its environment without any problem. When you have multiple cells in a structure like this, you have cells on the inside and you have cells on the edge. And so the cells on the inside can't get access to things on the edge, such as nutrients and oxygen. And so the cells on the inside are either going to maybe die, or they're going to evolve into something that's, very alien to the other part of the organism, or there's going to be some sharing of resources across all these cells. And so this is why we have circulatory systems or other types of systems where nutrients are transferred, like in plants. There's a lot of nutrient transfer up and down the root system. And so you'll see this in a lot of multicellular organisms where you have to share resources across the colony. And so Euler circuits, in a very technical sense, are an attempt to cross every independent edge in what we'll call multi-cell network. But it's basically some network where you visit every edge once and only once. And the idea is that you don't want to have any self-crossings. So you don't want to visit any cycles or any edges twice. You only want to visit all of them once, and that's it. And the reason I introduced this constraint is because it's something that we understand mathematically, but also because you can imagine in terms of symmetry, if you visit a bunch of edges multiple times, that gives those edges an advantage in terms of flows and other things. And so we'll just use these constraints, and you'll see how this works in a bit. So for a network of triangles, you have a symmetrical transformation. You can, divide them. We'll take a network of triangles, for example. You can have a symmetrical transformation where you go from one triangle to two triangles, and you end up with either an odd number of cells or edges or an even number, and then that has an effect on whether you can complete an Euler circuit. So I think I should just get on with the graphical part of this because it's more informative. So you have this. There was this program called Graph Online I was using to generate these. So this shows you, like in a geometric network, what kinds of shortest paths you can generate. So we have these nodes and we have these edges. The edges are in yellow, the nodes are in red, and the nodes are sort of the end of each edge and where they intersect, and the edges are the flows across this network. So we have this graph actually exhibits a Eulerian path. This program will find a Eulerian path because, as it turns out, Eulerian paths, just like the TSP, is NP hard, and it's hard to get an analytical solution just by hand. In this example, it's a lot more complex. This does not have a Eulerian path. So this is a combination of triangles and rectangles, and this configuration does not have a Eulerian path. That's the important thing to remember. And the number of edges, I think, is odd. But I haven't been able to figure out any interval where you have Eulerian edges versus not, or a Eulerian path versus not a Eulerian path. It's not as simple as the even-odd distinction I mentioned earlier. It's something else. It's where you take, like, say, a triangle and you divide it into two triangles, and that's a Eulerian path. But then when you introduce maybe a fourth or fifth triangle, you don't get it. And then if you introduce a rectangle, you'll get it back. And so as you grow these structures, you run into situations where you can no longer build a Eulerian path. And then if you add different shapes or different, maybe another cell division, you can get it back. And so, but that's not the only thing we can find out from this exercise. We can do things like rotation, scaling, shear, and perspective of these different shapes. So we don't just have to deal in terms of these very rigid looking structures. We can build things that look more biological and have more difference of shape in them. And of course, we can conformally map like these nodes so that we can stretch them out. And so this is like development, where you're taking like a landmark in an embryo or in a larval organism, and you're stretching it in certain ways. And so this is a conformal mapping showing these dots, and this would correspond to these nodes. And then you're stretching this node that was up here, you're stretching it out like this, but you're able to retain this Euler path. I think we had a question in the chat, but let me go check it out. Oh, what are the numbers of the nodes mean? I think this is just the way it numbers them. So it numbers them in a way that's, I think it's arbitrary with respect to the program. So it just numbers them so it can keep track of the cycle. So the circuit would have like 1, 2, 3, and I think it's the way I put them on the board here. So they're, but the program keeps track of these numbered. So it like goes, the path will go from like 1 to 3 to 0 to 9 to 1. And so, it shows you up here, it just has an identity. And they don't really mean anything other than just an identity. But that's how this works. And so you can do a lot of things with this type of simple geometric approach and understanding how these things work. Now this is where it gets interesting, is that, and this is what sort of, this brings it closer to life. So this is very similar to like a lineage tree in development. So in this case, what we're doing is we're just simply using a replication rule, all right, in this case. And we're building, we take this triangle and we replicate it. And then we try to build one of these Eulerian circuits on this object that results. And so in this triangle, we can build an Eulerian circuit pretty easily. And so it has a cost function of 0, cost function value of 0. You go down and you add a triangle to it like this, where you make this shape, and it has a cost function of -1. So now it's no longer completing that Euler circuit. So now you have a choice. You can either add something else to get back to the original Euler circuit, the complete Euler circuit, or you can add another piece that looks the same. So you can just have a simple addition of a piece, or you can add some new piece. And as we can see here, if you just do a replication of the same motif, you still don't recover your Euler path, but in this case you do. And so the idea is that you have these networks between on the outsides and on the edges of these cells that are equivalent in terms of their shape, in terms of their flow, in terms of their accessibility. And so that's there. And this is what I'm calling modularity. So you're actually creating a new module within the organism. So we talk about modularity in terms of maybe like a functional subdivision. And this is, of course, a module that's different from the rest of the organism. So you have this, again, you can't complete an Euler path this way. So you add another triangle up at the top, and you recover your Euler completeness. If you add another triangle in another direction, you actually score worse. But then if you add components inside these objects as they're replicating, you actually recover your Euler completeness. And this is again modularity because you're creating new modules, phenotypic modules. There's probably an analysis to be done on the phenotypic modules themselves, but I haven't done that on this. And so this actually resembles this is what Dick was talking about when he did his triangular cells talk. So this is a triangular cell. So, cells can take on a lot of shapes. If you look in human cells, they take on a lot of shapes, but especially like in algae and in microorganisms, they have a lot of shape diversity. And so this is one way we can simulate this. Again, we can do this with hexagons. With hexagons, it's interesting because if you use a simple replication rule, you always get Euler completeness. And that's interesting because one of the hypotheses about honeycombs, the structures that honeybees build to store honey and things like that, is that they're optimized for packing in a maximal number of cells in a small space. And so this kind of suggests that that's correct, that there's some optimality to hexagons. And so I haven't analyzed these any further, but this is something that is an interesting result. So there's no lineage tree here. It's just kind of a progression of replication of cells. And then, of course, you can use, this is mess. This is not supposed to be here, but this is a very complicated lineage tree here, where you have this square, and then you divide it into two squares, and then you can continue with the square motif where you're adding in. You just have a simple square replicator that continues down this path, and it keeps producing squares in this grid, and that also shows this serial Euler completeness. So I call it fractal reproduction because you can reproduce as many squares as you want, or rectangles as you want, and always get the same result. It never gets broken. And so there's a fractality to this. There are no modules in these. In this case, you have an asymmetric transformation. So say you had a rectangle of a different size. In this case, you would have an asymmetric transformation that still preserves that Euler completeness. You can then divide this bottom cell up, so it can divide and take on something like a grid like this, and that preserves its Euler completeness. But if it's off center, if it's an asymmetric division, then it breaks that Euler completeness, and you can continue down that road of no modular. There's no modularity, nor is it like a complete Euler cycle. And so that might be a non-viable phenotype. In this case, we have modularity, but then if we add in like a triangle at the bottom, you can't see it here. But if you add in a triangular motif, then you get this -1, and you'll get modularity because you can recover the Euler completeness. So those are some of the experiments I've done on that. And this is, again, another cell lineage tree where we're just using different rules, and we're getting different, we're able to recover Euler completeness in a lot of these structures, or sometimes we don't. And so those, I think they correspond to non-viable lineages, or places where you get modularity, which is like this functional subdivision or whatever. It's still, I've had a lot of trouble kind of defining the conditions of the rules in terms of replication, or when do you switch to a new shape, or things like that. Those are hard to map to the biology, but I think this is just the same thing. But I finally wanted to end with this mention of on growth and form. So this is a book that was written by Darcy Wentworth Thompson, who is a morphologist and a developmental biologist about 100 years ago or more. He wrote a book. The 100th anniversary of his book was, I think, last year, where it's a thick book. And in this book, he talks about sort of the mathematical structure of seashells and other organisms. He's famous for using mathematical transformations to look at different phenotypes in fish. So you have these different fish that you find, and the idea is you take a single fish, a bony fish, and you put this grid over it. and you mark the landmarks in different parts of the grid. So different intersections of the grid correspond to different landmarks. And then when you put a different fish on the same grid, those landmarks are misaligned. So you realign the grid according to where the landmarks have moved to. And so you can create these warped grids that actually describe changes in phenotype across fishes. They might be related phylogenetically, or they might be very different. But I think in light of what we were talking about, machine learning and feature selection, that this is an interesting thing that was done about 100 years ago. And when he did this, he didn't have like the benefit of supercomputers. He had to do it by hand. And so this is a really, I think, a remarkable feat considering. But we don't really understand a lot of what went on in this book, other than like just kind of what he displayed. I mean, people have tried to do computational forays into some of this. There's a book on growth forming computers, which is about 20 years old now, and it was an update to this. And they did a lot of morphogenesis in computers, but no one's really, I mean, I don't think people have looked at like the biological significance of a lot of it. And so that's my talk on that. I thought that would be a good talk to revisit in light of, thank you for the +1, in light of Dick's talk a couple weeks ago. So I just wanted to pull up that slide deck just to show people what was going on with that. I'm not sure where that work is going to go, but it's kind of like, I don't know how we're going to push it forward, but it's probably something. There's something there, I'm sure, but I don't know what it is. And then as for the on growth informed stuff, this is something that we can talk about further as well. There actually is a lot of like work that's been done between like mathematics and developmental biology, but it was done a long time ago, where there's a lot to follow up on that people haven't necessarily done. And Dick knows a lot about that. So when he's in the meeting, maybe we can ask him. I don't know. But so are there any other comments? Mayukh? Oh. Okay, so if there are any other comments?
Yeah, could you just share the slides? I just want to take a good look at the Euler paths thing.
Oh yeah, I'll share the link, and then if you can't get
it's kind of interesting.
Yeah. Just ask for permission if it doesn't share right. But yeah, so take a look and see what you think. If you have comments, please give them, and that'll be good. Okay, so thanks for attending this week. Next week we'll have another set of updates on open or on GSoC. We'll have maybe some other things as well, maybe some other presentations. If you have any questions in the meantime, please contact me on Slack or or email. So thanks for attending. Have a good week. All right. Bye. Stay safe.